All right, here is the quick lowdown on your lab. Basically, read the directions super carefully. You are doing two things. You're going to make a little section on all the stages of the cell cycle, and then you're going to do some math and do some calculations on like percent of cells in mitosis. So, a couple things. You're doing this on a blank doc. Not a Google slideshow. So set your margins small, 12 point or smaller, put a title, etc. So read all those directions. One thing I do want to emphasize is number 3. Make sure that images are clearly labeled, that sort of thing. You are going to be zooming in beyond what you do in your pictures. So essentially, I want you guys to find like the best example of metaphase, for example. And in your cell phone, I think it's easiest to do in your cell phone, is you'll zoom in on that cell and zoom in until you lose resolution. Once you lose resolution, zoom back out a little bit and make sure that image is perfectly crisp and clear for that cell in metaphase. There's still going to be other cells around it. That is totally fine. You just want to zoom in as much as you can while still maintaining a crisp, sharp image. Then crop it, right? And then how will I know which cell you're talking about? Well, you're going to label it. So I'll be able to see which cell you're using as your example for metaphase. Remember, your job is to find the best examples. So take good pictures, take a bunch of pictures in case like you're like, oh, shoot, this metaphase is actually not that great. So you have options to choose from. Make sure to follow directions, all that stuff. You have to identify the source of each photo. So like cite your sources, even if you're the one taking the pic, say like photo by Sarah Bell. That way I know who took the picture. Even if it's your lab, if you forget to do that, I'll assume something bad. So just don't do that. Okay. For the cell cycle part, obviously we are using the root tip. Like I said, zoom in as much as possible. Don't worry about the arrow part. If the arrow isn't pointing to the cell in question, that's fine because you're actually going to be zooming in anyways and probably using different cells within one picture. Label any structures you can actually identify. Describe what you see that is happening in the photo. So remember, descriptions are not the same thing as labels. Like I wouldn't have nucleus breaking down as a label. That belongs in the description. So labels and descriptions are different. And I'm not going to describe something under the actual photo of something I can't see, right? So like if I say something about ribosomes, obviously that does not belong here. The second half of this is on the right-hand side, you're going to do Google, use Google, find a good diagram of a cell in interphase, like a diagram, a cartoon, and that one can be pre-labeled. You'll cite your source, and then you will, oh, I don't know if you guys can see this, but my stupid later. And then you'll cite your sources and describe everything else that you can't actually see happening in the photo. Okay, so this is like real life, what we can see. And then this is like what we know is happening, but our microscopes are not good enough to pick up. That's basically part A. Do one cell for interphase. So one cell for all the stages of interphase is totally fine. Then do one cell for each of the stages of mitosis and make sure that you do a cytokinesis and telophase separately, even though in real life they occur together. I want your descriptions of telophase and cytokinesis to be separate. So ideally you'll find a cell in like maybe early telophase and then a cell in like late cytokinesis for your pictures. And then the descriptions should be pretty easy. So I want separate boxes for telophase and cytokinesis. I've now said that twice. Okay, the mitotic index, follow the directions. Take a picture, get a rough estimate of the number of cells in that picture, and then count the number of cells in prophase, metaphase, anaphase, telophase / cytokinesis, those ones you can lump together here, and then calculate how many or the percent of cells in interphase, mitosis / cytokinesis, and then use your book to figure out mitotic index. It's not hard. Show your work. Give me the picture. And don't forget, remember, anything you do that is beyond sharing a raw, unedited, uncropped, unzoomed, unlabeled photo, that is the only thing you can do within this lab that is academically integritarily, is that a word? Okay. Anything past that, nope, we're not doing that. Don't try me. I have a really good memory and I remember things. So I catch them. It's best to do this on your own. Feel free to email me this question or email me this weekend if you have quick questions, and follow directions. Keep working on your study guides, and I will see you guys soon.